Okay. So, hi everyone. I think so. I am live. I am so sorry for the delay, as I always make. So, but basically today I was on time at 6:25. I opened my laptop and I was about to go live, but my camera was not working. Now is it fine? I can see so many chats. मैं थोड़ा chats पढ़ लूँ क्या? मुझे ऐसे chats पढ़ने में बड़ा मजा आता है. Shivesh is here, Shri Shai is here, okay, Sid is also here. Hi. That's great. Okay, who wants to increase the size? Good. Complaint पियो, size बढ़ेगा. Scam था ना ये? Scam था ना? बचपन में complaint पिया था क्या तुमने? हाँ? चलो, okay. Let's start the session बच्चों. Let's start the session. अच्छा, session का thumbnail क्या है? हाँ ठीक है, DNA replication पढ़ाना ना मुझे. चलो, मैं पढ़ाना start करूँ. Should I start? Should I start everyone? Okay. So complaint से कुछ भी नहीं हुआ. मैंने भी पिया था, मैं भी छोटा ही रह गया. I think so. My height is around 6 feet तो नहीं, 6 feet से छोटा ही हूँ मैं. चलो, let's start the session बच्चों. Let's start the session. See, so today what we are going to do is that we are going to try to learn about the DNA replication, like how the DNA replication is taking place, how the DNA replication is taking place, and how process is going on, what enzymes are needed, all those things we are going to study. Okay, clear? Okay, you are 5'10", maybe approximately. I'm also 5'10". I don't remember my height surely. Okay, chalo, let's start. So before that I'm going to teach you something which is known as central dogma. Okay, I'm going to teach you something which is known as central dogma. So guys, what is central dogma? Anyone, any idea about the central dogma? What is it? What is it? Sid, long time Sid है ना, बहुत दिन बाद आए हो Sid मेरी क्लास में. Central dogma. Okay, we are going to talk about the central dogma. So what is central dogma, sir? See here. First of all, what happens na? Let's talk about the suppose that there is a sperm. Okay, there is a sperm. ये किसी का sperm है. And then second thing is that someone's ovum is there. Okay, someone's ovum is there. Now this sperm and ovum will get fused and they will going to make what? They are going to make zygote. Yes or no? Yes, obviously so. Now this zygote is going to make what? This zygote is going to make us. We have also being made from the single cell zygote. So let's write here baby. Okay, baby is produced. Clear to everyone? Yes. Synthesis of protein. Yes, exactly. Baby is produced. Okay, now the person who was having this sperm was, let's suppose, having height like 6 feet, was 6 feet tall. Okay, so the baby was also 6 feet tall. Just suppose, just for an example, the baby was also 6 feet tall. So how this information from the sperm went to this baby? Sperm तो खुद छोटू सा होता है, इतना छोटा होता है बेचारा, sperm का तो कुछ height ही नहीं होता. But still this sperm is able to what? This sperm is able to transfer the information from itself to the baby and baby became 6 feet. Not baby, बच्चा ही 6 feet का पैदा नहीं होगा, मतलब बड़ा हो गया तो 6 feet का हो गया. Yes or no? Right? तुरंत थोड़ी पैदा हो जाएगा, वो हुआ बस एकदम अभी हुआ है बस 6 feet का हो गया. नहीं, it will take time, but maximum height this baby acquired was 6 feet. Okay, so how does this happen? See here. So in sperm, what was there? In sperm, if I talk about the structure of the sperm, so somehow sperm looked like this. This is known as the head of the sperm. Yes or no? Yes. Inside the head of the sperm we have the nucleus, right? We have the nucleus, and then what? Inside the nucleus then we have the DNA. Yes or no? Inside the nucleus we have the DNA. In the similar manner, guys, in the similar manner, this was the ovum. This was the ovum. Yes or no? Yes or no? And if I talk about this ovum, inside this ovum also we will have nucleus and nucleus. Inside the nucleus, तुम ऐसे कमेंट करते हो ना तो मैं कमेंट देखने में लग जाता हूँ. चलो इधर फोकस करो, ठीक है? So inside the nucleus what do we have? We have the DNA. Okay, right. So both sperm and ovum was having DNA. Yes or no? Yes, Shrishai Sivesh. Both sperm and ovum was having the DNA. Now, this DNA is having all the characters. This DNA is having all the characters. Yes or no? तुम कैसे होगे? तुम मोटे होगे, पतले होगे, बुद्धू होगे, इंटेलिजेंट होगे, या तुम बड़े होगे, छोटे होगे, तुम्हारा बाल भूरा होगा, गोल्डन ब्राउन होगा, ब्लैक होगा, तुम्हारा स्किन कलर गोरा होगा, काला होगा, whatever the information will be, all information will be there in the DNA. Yes or no? Now this DNA will do what? This DNA will make the RNA. From this DNA, the RNA will be made, and from this RNA, a protein will be made. So this protein will be responsible for all the characters. Like this protein is responsible for all the characters to be expressed. Characters है किसके पास? DNA के पास, right? कैरेक्टर्स है किसके पास? डीएनए के पास. बट एक्सप्रेस कौन करा रहा है? एक्सप्रेस करा रहा है भाई प्रोटीन. एक्सप्रेस कौन करा रहा है? प्रोटीन. इट्स सपोज लाइक दैट माय मैनेजर told me that, sir, you have to take this class. Okay, so my manager ordered me something and I am taking the class. Okay, so DNA order दिया RNA को, RNA ने प्रोटीन बनाया. Clear to everyone? Yes or no? Any doubts? And this is known as the central dogma. This is known as the central dogma. Yes or no? चलो now इसको थोड़ा सा अच्छे से लिख लूं. We have this DNA. We have this DNA, and then we have the RNA, and then we have what? And then we have, ये तुम्हें बम की आवाज आ रही है? बम फोड़ रहे मेरे तरफ ये लोग. DNA, RNA, and then we have the protein. DNA, RNA, and then we have the protein. Tarun sir is best. Yes, Tarun sir is best. No issues in that. He is a very good teacher. Chalo. Okay. Now we have this DNA, then RNA, then protein. Okay. Now what is going to happen? DNA से RNA बनेगा. When DNA से RNA is being made, when DNA to say RNA is being made, what is going to happen? Transcription is going to happen. What is going to happen? Transcription is going to happen. Yes or no? Transcription. Yes or no? Okay. DNA से RNA बना, then the process is known as transcription. Now from RNA to protein is being made, the process will be called as translation. The process will be called as translation. Yes or no, guys? Yes. Shivesh, Shrishai said yes or no? Yes or no? Any doubts? Hi Midlife, how are you? Any doubts? Let me know. कोई डाउट नहीं है. ओके, so from DNA to RNA transcription और from RNA to protein अगर बना रहे हैं तो the process will be called as translation. Now what, sir? Now see, sometimes what happens, DNA has to be replicated, right? मान लो तुम्हारा हाथ में कुछ सेल्स टूट गए या हाथ टूट गया या कुछ भी रिपेयरमेंट की जरूरत पड़ी, सो वहां पे क्या होगा? नई डीएनए का जरूरत पड़ेगा, नई सेल का जरूरत पड़ेगा. सो समटाइम्स व्हाट हैपेंस, डीएनए आल्सो गेट्स रेप्लिकेटेड. यस और नो? सो डीएनए से डीएनए बना रहे हो, देन व्हाट? सो डीएनए से डीएनए बना रहे हो, देन व्हाट? डीएनए से डीएनए बना रहे तो कौन सा प्रोसेस? दैट प्रोसेस इज नोन एज डीएनए रेप्लिकेशन. That process is known as DNA replication. Okay. Why we directly not make protein from DNA? See, DNA is not a very good structure. ये कम्युनिकेशन डीएनए का अच्छा नहीं है. सो डीएनए क्या करता है? पहले आरएनए को कमांड देता है, फिर आरएनए जो है वो प्रोटीन बनाता है. डीएनए कभी डायरेक्टली प्रोटीन नहीं बनाएगा, बिकॉज डीएनए एक तो डबल स्ट्रैंडेड है, कम्युनिकेशन अच्छा नहीं करता. तो इंफॉर्मेशन का ट्रांसमिशन अच्छा कौन कर रहा है? आरएनए. तो ये काम आरएनए ही करता है. यस और नो? ओके, क्लियर टू एवरीवन? सर, इज द प्रोटीन शेक वी टेक इज ट्रांसलेटेड एक्सटर्नली? नो गाइस. प्रोटीन शेक विच यू टेक इज बेसिकली व्हाट? हम प्लांट का प्रोटीन एक्सट्रैक्ट कर लेते हैं. प्लांट में जो प्रोटीन है उनको एक्सट्रैक्ट करके तुम्हें पाउडर बना के दे दिया जाता है. यस और नो? अगर प्लांट बेस्ड ले रहे हो तो प्लांट का प्रोटीन को प्रोसेस करके तुम्हें दे दिया जाएगा. अगर हम एनिमल बेस्ड प्रोटीन ले रहे हैं तो सेम चीज, उनको ड्राइड किया जाता है, बोन्स लिए जाते हैं, ड्राइड किए जाते हैं, उसके बाद हमें दिया जाता है. यस और नो? इट्स नथिंग रिलेटेड टू ट्रांसलेशन एंड ट्रांसक्रिप्शन. ओके, इट्स नथिंग रिलेटेड टू ट्रांसलेशन एंड ट्रांसक्रिप्शन फ्रॉम दैट प्रोटीन. ओके, क्लियर? सो इन टुडेज सेक्शन सेड छोड़ दो, सेड उसको छोड़ दो. ऐसे मेहमान आए हुए मेहमान को हमें ऐसे नहीं बोलना चाहिए, ठीक है? नो हार्ड वर्ड्स हियर, ओके? चलो चलो चलो, लेट्स फोकस। सर क्लास कितने बजे से स्टार्ट होती है? क्लास स्टार्ट होती है 6:30 से, बट दिस टीचर ऑलवेज कम्स लेट 5 मिनट्स, सो यू कैन हैव दैट 6:30 to 6:35 टाइम पीरियड। ये सोनू, तुम ना ऐसे गुस्सा तो नहीं करते मुझसे कि सर आप रोज लेट आते हो? थोड़ा बहुत मैं लेट आ जाता हूँ यार, क्या करोगे? चलो, let's start. So in today's session, what we are going to do is that we are just going to talk about the DNA replication. So first of all, I am going to make you clear that मैं सेमी कंजर्वेटिव का जो एक्सपेरिमेंट है वो आज नहीं कराऊंगा, ओके गाइज? मैं सेमी कंजर्वेटिव का जो एक्सपेरिमेंट है, आई एम नॉट गोइंग टू डू दैट एक्सपेरिमेंट। वो एक्सपेरिमेंट हम कल करेंगे। आज सिर्फ और सिर्फ समझेंगे कि डीएनए रेप्लिकेट कैसे होता है, सेमी कंजर्वेटिव नेचर एक्चुअली में होता है। क्लियर एवरीवन? एनी डाउट्स? श्रीशाई, शिवेश, लेट मी नो इन द कमेंट सेक्शन फटाफट से। शुड आई मूव फॉरवर्ड? great Shrishai, great. Chalo, done? Let's start. Okay, so now see what happens. One more thing I'm going to ask you, okay? Let's suppose how should I ask this question? See, okay, chalo, the question is, do we have same DNA in every cell? Do we have same DNA in every cell? Yes or no? Give me the answer in the form of yes or no. What do you think? Guys? कि तुम्हारे बाल में भी सेम डीएनए, तुम्हारे पैनक्रियाज में भी सेम डीएनए, तुम्हारे लिवर में भी सेम डीएनए? डू यू हैव सेम डीएनए इन योर थायराइड ग्लैंड? डू यू हैव सेम डीएनए इन योर ईयर? What do you think? Everything is having different function. You see, your liver is making something else, your thyroid gland is producing thyroid, your hair has different function. So what's happening? Like, all the cells are very different, but still, are they having the same DNA? Yes, exactly, Shrishai. Very good. Obviously, na, see, you have been produced by this single zygote. Yes or no? इसी zygote में तो division हो के तुम बने हो. तो अगर इस zygote में एक DNA था, तो इसी DNA को तो replicate कर करके we have been made. Yes or no? So DNA will be what? DNA will be same in all the cells, but few parts of the DNA will be activated in special cells. Like, let's suppose that if I am talking about the thyroid gland, and if I am talking about the skin, if I am talking about the skin, so thyroid gland is having what? Thyroid gland produces thyroid hormone. Yes or no? सबका जीनोटाइप सेम है, बट फिनोटाइप अलग अलग है. Yes, we can say that. Easy explanation, we can say this. Phenotype external, कुछ characters अलग हो जाते हैं, right? See, I'll make this thing clear, very clear, okay? Thyroid gland is what basically? Thyroid gland produces thyroid, yes or no? Thyroid gland produces, sorry, क्या बोल रहा हूँ मैं? Thyroid gland produces thyroid hormone, okay? Thyroxine. But your skin doesn't produces that. Your skin doesn't produces that. Why is it so, guys? Because in thyroid gland, वो thyroid gland में thyroxine gene activated और तुम्हारे skin में वो gene activated नहीं. That means what? In all our cells, some genes are activated and some genes are not activated. See, if I talk about your hair, so in hair keratin is produced, in nails keratin is produced, but हाथ में keratin produce नहीं हो रहा, तो यहाँ पे keratin वाला gene active है, यहाँ पे keratin वाला gene active नहीं है. Yes or no? And this is the reason why all our cell is not very similar, right? In spite of having the same DNA, हमारा सारा का सारा cell अलग अलग है. Yes or no? Because किसी में क्या हो रहा है, कुछ part of gene activated है और किसी में कुछ part of gene activated है. See, is this clear? Is this clear to everyone? Yes or no? Any confusion? Should I explain it again? देखो, simple, midlife बहुत simple है. तुम्हारा जो बाल का जीन है, your hair is, okay, I'll take the example of thyroid gland. Your thyroid gland is what? Your thyroid gland is producing thyroxine hormone. Yes or no? Yes, okay. And your skin is not producing the thyroxine hormone. Yes. So in thyroid gland, the cells of thyroid gland is having a gene of thyroxine hormone which is active there. But in skin, that gene is present, but that gene is inactivated in skin. Is it clear, midlife? Any doubts? Explain everything. Sorry, easy explanation, Shivesh. Any doubts anywhere, let me know, okay? Any doubts anywhere, let me know. Clear to everyone? Okay? Any doubts anywhere, let me know. Everyone. इजी सॉरी इजी एक्सप्लेनेशन, इज इट क्लियर? यू वर राइट, ये जो फिनोटाइप वाला था यू वर little bit right, but this is the correct explanation. Like in some cells some genes are activated and in some cells those genes are inactivated. Yes or no? Clear? Okay, great. Now अब हम start करते हैं अपना असली काम. What असली काम? That means DNA replication. Okay, I'll promise you guys you won't have any doubts in DNA replication. सारी चीजें मैं यहीं याद करा दूंगा. Okay, okay, Sania, yes, this is good. Sania, this is good. Chalo. Now what? Okay, we have two organisms. I'm taking two organisms. One I'm taking E. coli. Okay, one I'm taking E. coli. Okay, guys, E. coli is a prokaryote or a eukaryote? E. coli is a prokaryote or eukaryote? जल्दी बताओ मुझे. Whether this E. coli is a prokaryote or eukaryote? Prokaryote, exactly. It is prokaryote. Yes, Sid. So this E. coli is prokaryote. This E. coli is prokaryotes. Okay. Oh, sorry, eukaryotes. बच्चों क्या तुम भी ना हैं? ये E. coli prokaryote थोड़ी है. Prokaryote है क्या E. coli? भाई eukaryotic cell है. Eukaryotic cell है. Sid, eukaryotic cell है ना prokaryote. हाँ? E. coli prokaryotic है क्या? यूकैरियोटिक है ना बच्चों. ई कोलाई यूकैरियोटिक है प्रोकैरियोटिक है क्या है? 1 सेकेंड बच्चों, 1 सेकेंड रुक जाओ, तुम लोगों ने ना मुझे भी कंफ्यूज कर दिया. 1 सेकेंड, ये क्या हुआ? 1 सेकेंड, सॉरी, ई कोलाई प्रोकैरियोटिक है ये. I'm so sorry. E. coli is a prokaryote. Yes or no? I am confused. I'm so sorry. Said you are correct. Said you are correct. Monin, yeah. Okay, ठीक है, चलो. So if I talk about the eukaryote, if I talk about the eukaryote, E. coli. So it's what? It's a bacteria. It is a bacteria. And if I talk about this bacteria is what, guys? This bacteria is prokaryote, not eukaryote. Okay, this bacteria is prokaryote. And it is going to have what kind of DNA? It is going to have what kind of DNA? Circular DNA. Yes or no? Prokaryotic है बच्चों. Sid was right. It is going to have what? It is going to have a circular DNA. Yes or no? I am so sorry. Circular DNA. Yes, Shrisha. Yes. Circular DNA. कैसा DNA होगा इसका? Circular DNA. Yes or no? On the other hand, let's take the human beings. Let's take the human beings. Yes or no? Yeah, let's take the human beings. So human beings is having what kind of DNA? Linear DNA or tell me, human beings is having linear DNA or what kind of DNA, guys? Linear DNA या circular DNA? Human beings के पास कैसा DNA बच्चों? Human beings के पास कैसा DNA? Human beings के पास कैसा DNA? Linear है या circular है? So we have a linear DNA. Yes or no? Yes, exactly. We have a linear DNA. Yes or no? Great. Midlife, Sania, linear DNA है, सबको को पता है. dsDNA is double stranded linear DNA. Okay, now what we are going to do is that we are going to study the DNA replication in both of them, but DNA replication process in both of them is similar. DNA replication is similar in both of them. Replication is similar in both of them. So what we are going to study anyone and दूसरा में भी DNA replication बिल्कुल वैसे ही का वैसे ही होगा. Clear to everyone? Okay, start करूं क्या? Should we start DNA replication? Everyone ready? बिल्कुल concentration यहां होना चाहिए, ठीक है? Okay, चलो. Now I am going to explain you the word semi-conservative first. Okay, so see what happened na. There was a couple. Okay, there was a couple. एक couple था, ठीक है? Couple means what, sir? Girlfriend, boyfriend, obviously. So चलो, ये दोनों क्या है? They are the girlfriend, boyfriend, right? ऐसे हमारे स्कूल में एक होता था ना, like पूरे स्कूल को पता होता था except that teacher. So these are what? These are girlfriend, boyfriend. But what happened? इन दोनों में बनी नहीं, तो ये दोनों क्या हो गए? अलग अलग हो गए. ये दोनों क्या हो गए? अलग अलग हो गए, ये इधर आ गया, ये इतना दूर चला गया. ओके, ये दोनों क्या हो गए, दे बोथ आर सेपरेटेड नाउ. नाउ व्हाट हैपेंड टू दिस गर्ल, this girl met another boy and this boy met another girl. This boy met another girl. So what is happening? दो डीएनए था, दोनों अलग हो गए और एक डीएनए के पास दूसरा डीएनए बन गया और दूसरा डीएनए के पास, ये वाला डीएनए के पास अगेन दूसरा डीएनए बन गया. yes or no? yes or no? is it clear? is this thing clear? बताओ, is this thing clear? right? So जब भी नया डीएनए बन रहा, whenever the new DNA is being formed, a part of old DNA is present, a strand of old DNA is present, and such type of method is known as semi-conservative method. Semi-conservative method. Why is it always like this? What, Shrishai? Like this method? See ना, if semi-conservative will not be there, so copy कैसे करोगे तुम? ये डीएनए से, ये डीएनए है, इसमें जो कोड्स है, उसको कॉपी करना है हमें, राइट? सो अगर पहले वाला नहीं रहेगा तुम्हारे पास तो तुम नया वाला को कॉपी कैसे करोगे, किसके बेसिस पे कॉपी करोगे? यस और नो? यस और नो? मिडलाइफ, सिड, शिवेश, यस और नो? यस और नो? क्लियर? ओके, ग्रेट. शुड आई मूव फॉरवर्ड? ओके, चलो. हाँ, बस वो गर्लफ्रेंड बॉयफ्रेंड वाला अब यहीं तक छोड़ देना, अब उसको वहीं छोड़ देते हैं, अब आगे बढ़ते हैं. यस और नो? गंधर्व बाबर, गुड, वेरी गुड, नाइस. आई एम अ लवेबल टीचर, राइट? यस और नो? चलो. सो दिस इज व्हाट, दिस इज क्लियर, सेमी कंजर्वेटिव ऑफ डीएनए. अब देखो, अब क्या होता है, कोई भी एक डीएनए को हम उठाते हैं. नाउ लेट्स चेक हाउ द डीएनए रेप्लिकेशन इज गोइंग टू हैपन. How the DNA replication is going to happen. Let's check that. How the DNA replication is going to happen. Okay. So see here. First of all, we are going to have what? We are going to have a linear DNA. क्या है हमारे पास? एक linear DNA है. So let me make a linear DNA first. Okay. Right. Now, what is going to happen? The replication has to start somewhere. तो तुम खुद सोचो, इफ यू हैव अ वॉल इन योर हाउस और उसको तोड़ना है, ठीक है, तो तुम कहां से तोड़ने की कोशिश करोगे? जहां से वो वॉल बहुत मजबूत होगा वहां से, या जहां से वॉल बहुत छोटा होगा वो? यस और नो? यस और नो? यस और नो? कहां से तोड़ोगे? वहां से तोड़ना चाहोगे फ्रॉम वेयर द वॉल इज वेरी वीक. यस और नो? ओके. सो DNA replication starts from where? It starts from AT rich site. It starts from AT rich site. Now why guys? Why DNA replication starts from AT rich? Why DNA replication starts from AT rich? Why DNA छोटा weak Sanya weak, right? Chota you mean weak, okay? So why it starts from AT rich, guys? Because in GC rich we have 3 bonds pair. Yes or no? In GC we have 3 bonds. In GC how many bonds we are having? 3 bonds. And in AT, between A and T we are having 2 bonds. Yes or no? Yes or no? So the DNA replication will start from this area. Okay, right? T A T A T. I have just written randomly. Exactly, AT rich side. No issues, Shrishai. अभी सुन लो, अभी याद कर लो इसको, ठीक है? Everything I am gonna cover it. Your molecular basis of inheritance will be very much clear. That's my promise. अगर फिर भी नहीं होगा तो फिर पढ़ा दूंगा मैं, क्या दिक्कत है? Now what is going to happen? A protein will come. What? A protein will come, which is known as DNA A protein, which is known as DNA A protein. Which protein is going to come? DNA A protein. Clear? कौन सा प्रोटीन आएगा? DNA A protein. This protein is something like this. This protein is something like this. It will come here and it will make a hook. It will come here and it will make a hook. Which protein is coming? DNA A protein is coming and it is making a hook. Now once the hook is made, ये क्या करेगा? DNA को ऐसे पकड़ेगा दोनों तरफ से और ऐसे खींच देगा. क्या करेगा? ऐसे पकड़ेगा और it will pull the DNA. Okay, so something like this structure will be made. Yes or no? Somewhat this structure will be made like this. Yes or no? This structure will be made. ओके समझने की कोशिश करो, कुछ ऐसा स्ट्रक्चर बन जाएगा। अरे हाँ या ना बच्चों, कुछ तो बोलो। यस और नो? Okay, great sir, great. Now this structure is known as what? This structure is known as replication fork or replication bubble. This structure is known as what? Replication fork and or replication bubble. हालांकि when you will go in higher classes now, so this replication fork and replication bubble is also a different thing. But for now we can remember that replication fork बोल दो या replication bubble बोल दो. Yes or no? Yes or no guys? I don't know why I'm hearing everything for the first time. I will fully concentrate. Okay, no issues. जहां पे ना समझ में आए, I am there. Let me know in the comment section. Okay, let me know in the comment section. कुछ एक्स्ट्रा नहीं पढ़ाऊंगा मैं. Whatever is given in the NCERT ना, I'll just read. I'll teach that only. So अभी तक क्या हुआ? Till now what we have seen, semi-conservative of nature of DNA. Semi-conservative nature of DNA we have seen. Now there is a protein which is called DNA A protein. वो आया, that came. Where it came? It came to 80 rich site. It came to 80 rich site and it get attached and pulled the DNA and a replication bubble or replication fork is created. Yes or no? A replication bubble or replication fork is created. Now the point where this DNA A protein came is known as what? Replication site. Initiation of replication. Initiation of replication. यहां से क्या हो रहा है? यहां से replication start हो रहा है. Yes or no? Here replication is starting. Now this site is the only site which is also known as ORI site. ORI site. नाम सुना है क्या तुमने? ORI site. नाम सुना है क्या? Yes or no? Let me know guys. Let me know guys. नाम सुना है क्या? Have you listened this name? ORI site. ORI site is what? ORI site is the point from where your DNA replication is gonna start. Now one more thing I am going to add here. See what's happening now. DNA of E. coli is very small. Yes or no? DNA of E. coli is small, right? Yes, in biotech, exactly. So अब origin of replication क्या है? Basically जहां से from where the replication is going to start, that is known as origin of replication. And what is it? That site is 80 rich. Okay. Now E. coli DNA is small. And meanwhile, if I talk about the human DNA, it is big. Yes or no? So see here, if DNA will be small, so ORI site will be what? Less. छोटा डीएनए एक जगह पे रेप्लिकेशन स्टार्ट करो, पूरा करवा लो, ठीक है, राइट? सो नंबर ऑफ ओरी साइट विल बी लेस. बट इफ यू हैव अ ह्यूज डीएनए, तो अ सिंगल ओरी साइट विल नॉट वर्क. We should have multiple ORI sites. Yes or no? So ORI site will be more. So that means I can say if DNA is what? Large, DNA बड़ा है, ORI site will be more. Yes or no? If DNA is more, then ORI site will be also more. If DNA is short, then ORI site will be short. Yes or no? Hi सार्थक, how are you? सार्थक, मैंने तुम्हें देखा नहीं. सार्थक कैसे हो? Is it clear everyone? Yes or no? बच्चों बताओ, फटाफट से बताओ. This concept is it clear? Right? तब तुम्हें समझ में आ रहा होगा कि क्यों E. coli have less ORI sites and why human DNA has lots and lots of ORI sites. Okay? Now again till now what we have read, my goal is for this session, my goal is that when we complete this session, DNA replication is very much clear to you. So I will be repeating things again and again. So please don't get irritated. You just listen and revise. Okay? I hope that works for you, everyone. See, till now what we have learned? We have learned that DNA was semi-conservative. And what happened when the DNA, the linear DNA, there was a protein called DNA A. It came and what happened? They pulled the two strands of DNA and replication fork or replication bubble is formed. Clear everyone? Okay. ORI site क्या होता है? जहां से replication start हो रहा है. देखो यहां से क्या हो रहा है, यहां से हम रेप्लिकेशन स्टार्ट करेंगे. अभी सानिया यहां से हम अभी रेप्लिकेशन स्टार्ट करेंगे, तो जहां से रेप्लिकेशन स्टार्ट हो रहा है वो हो गया ओरी साइट. ओके, क्लियर? सर, डज लॉट्स ऑफ ओरी साइट मींस मोर देन वन ओरी? यस, एग्जैक्टली, यस. ओके, चलो Now I'll be moving forward. I will be moving forward. So see here, now what's happening? They were boyfriend girlfriend. You remember that? ये क्या थे? ये दोनों क्या थे? बॉयफ्रेंड गर्लफ्रेंड थे. इनको अलग करना है. So ऐसा होता है ना, when they are like, when they have decided that okay, now we will be no more, we are going to break up. So what is going to happen? There will be some emotions, and that emotions, from that, on the behalf of that emotions, they will be like, चलो यार फिर से एक हो जाते हैं. So we have to prevent that. We have to prevent that. इतना मुश्किल से DNA A प्रोटीन ने इनको अलग किया है. इतना मुश्किल से DNA A प्रोटीन ने इनको अलग किया. बेचारे फिर वापस जुड़ जाएंगे तो क्या फायदा? तो here is one protein which is known as SSB protein. Here is one protein which is known as SSB protein. So this SSB protein do what? This SSB protein prevents the rebinding of the DNA strands. What it do? Prevents rebinding of DNA strands. Okay, of DNA strands. Clear? Any doubts? Breakup. Yes, breakup हो गया. Any doubts guys? Any doubt? This SSB protein, ये SSB protein क्या है? ये चाहता है कि इन दोनों वापस से कभी ना मिले. Okay, clear? Yes or no? Okay, great. Now what? I am making the diagram of NCERT. I am trying to make the diagram of NCERT. My diagram is very good, as you all know. So थोड़ा सा उसका replica बना लूं. So in NCERT they have made like this. In NCERT they have made like this. Yes or no? Now here we will start the replication. Should I revise you once again? Guys? एक बार फिर से रिवाइज करवा दूं क्या? चलो करवा देता हूं. मेरे लिए कर लो रिविजन. ओके, सो फर्स्ट ऑफ ऑल वी सॉ दैट there was two strands of DNA. बीच में कौन आया? DNA A protein. DNA A protein, what they have done? They have pulled the two strands of DNA apart, break up करा दिया. Okay. Now what? Now these guys wants to rejoin again, but to prevent the rebinding of the DNA, what came? SSB protein came. What came? SSB protein came. Okay. So since now they are not joining again, so we will going to start the replication. So to start the replication, we have what? We have an enzyme which is known as DNA polymerase. Which is what? Which is DNA polymerase 3. Which enzyme? DNA polymerase 3. Which enzyme guys? DNA polymerase 3. अब किसको लाओगे? पहले तो you have brought DNA A, then SSB protein, then what? Then DNA polymerase 3. कौन आया अब यहाँ पे? DNA polymerase 3. Okay. See, now this DNA polymerase 3 is what? Very nakhada wala enzyme. It is very nakhada wala enzyme. It is very nakhada wala enzyme. Single site binding protein, Sania. Single site binding protein. Okay. This DNA polymerase is what? It is very nakhada wala enzyme. Okay. DNA polymerase enzyme. Okay. My dog is also very excited. He's barking a lot. Are you able to hear his noise? DNA polymerase enzyme. इसका क्या क्या नखड़ा है? ये बोलता है मेरे को ये चाहिए, मेरे को वो चाहिए, मेरे को ऐसा कर दो, मेरे को वैसा कर दो, तभी मैं काम करूंगा, वरना मैं काम नहीं करूंगा. ओके, तो देखते हैं इसका क्या क्या नखड़ा. सबसे पहले तो इसकी प्रॉपर्टीज देख लेते हैं. इट इज हाईली इफेक्टिव. यस और नो? It is highly effective. See, अगर मान लो कोई बंदा आया तुम्हारे पास और तुमसे बहुत सारे डिमांड कर रहा कि मुझे ऐसा रूम चाहिए, I want AC, I want this, I want that, right? So why you will going to give? Because he is going to have some properties. Yes or no? Right? If suppose that you want to hire a maid for your house and that maid is very good, he or she is working very well, then you can give lots and lots of money. So first of all, that person should have a very good property. So here DNA polymerase enzyme is very nakhda wala enzyme, but it is having lots and lots of properties. It's highly effective. It is highly effective. It can replicate the E. coli's DNA within 18 minutes. 18 मिनट में E. coli का पूरा DNA replete कर देता within 18 minutes. How much? Within 80 minutes. Yes or no? Yes or no? Yes or no? Clear? Within 18 minutes, it is what? It is able to replicate the whole E. coli DNA. Okay? Clear? ओके, सो इट्स वेरी इफेक्टिव एंड इट डू दी प्रूफ रीडिंग. अगर मान लो चलते चलते कहीं इससे गलती हो जाती है, हालांकि इससे गलती होती नहीं, बट इन केस इफ इट मेक समथिंग मिस्टेक, सो इट कैन करेक्ट इट्स ओन मिस्टेक. ओके, सो इट डज प्रूफ रीडिंग ऑल्सो, इट डज प्रूफ रीडिंग ऑल्सो. दैट्स वाई ये इतना नखड़े वाला. अब नखड़ा क्या क्या है इसका देखो, इसका क्या क्या नखड़ा है वो भी देख ले. What does it says that? It says that I am always going to work from, I am always going to work from 5 prime to 3 prime direction. Always works from 3 prime to 5 prime direction. Now what is this 3 prime to 5 prime? Remember guys, remember we had studied that if one strand is 3 prime to 5 prime, the another strand will be anti-parallel, which will be 5 prime to 3 prime. Yes or no? And this guy, this DNA polymerase 3 always works from 3 prime to 5 prime. Always, I'm so sorry. Yeah, always works from 3 prime to 5 prime. मतलब कि अगर ये अपना DNA बनाएगा तो कहां से बनाएगा? See here. DNA कहां से बनाएगा? DNA यहां से बनाएगा. Yes or no? And if it is making the DNA will be what 5 prime to 3 prime. That means ये उस टेम्पलेट में काम करना चाहता है जिस टेम्पलेट का डायरेक्शन 3 प्राइम से 5 प्राइम है. Yes or no? Yes or no? उस टेम्पलेट से काम कराना करेगा जहां का टेम्पलेट क्या. yes, it is going to make from 5 prime to 3 prime, but template what? According to, with respect to template, it is 3 prime to 5 prime. With respect to new DNA, it is what? It is 5 prime to 3 prime. Yes or no? Yes or no guys? Let me know in the comment section. Sir, is it a catalyst? No, we can't say it's an enzyme. Enzyme is basically a kind of a catalyst only. Yes or no? Yes. Midlife clear? Midlife not. Sanya. Sid and Siddharth, what I am saying? Sid and Shivesh, is it clear? Is it clear? Okay, चलो कमेंट देख लो थोड़ा सा. Great. Okay, now this much is clear. अब आते हैं यहां पे. Now let's make the replication fork. अब थोड़ा सा replication fork पे आते हैं. Yes, midlife I am saying it's correct. See, नया DNA जो बनेगा ना सानिया, नया DNA बना रहा है वो 5 प्राइम से 3 प्राइम बनाएगा. कौन से टेम्पलेट के ऊपर काम करेगा? 3 प्राइम टू 5 प्राइम. सानिया सी, नया डीएनए जो बन रहा है वो कहां से कहां बनाएगा? नया डीएनए बनाएगा 5 प्राइम से 3 प्राइम, बट टेम्पलेट जिससे कॉपी कर रहा है उसका डायरेक्शन क्या होना चाहिए? 3 प्राइम टू 5 प्राइम. ओके, यस और नो? क्लियर? ग्रेट. अभी भी डाउट है सानिया, मुझसे पूछ लो. ऐसे डाउट लेके इस सेशन से बाहर नहीं जाने का. ओके, यस और नो? चलो. Now, see here, replication. Let's make the replication fork. So this is the replication fork. Okay, and this is opened. क्या ये खुला हुआ है? Due to the help of DNA protein A. Okay. Now see here, I am writing 3 prime to 5 prime. Here I am writing 3 prime to 5 prime. सानिया, is it clear ना? Clear है ना? कोई डाउट तो नहीं रह गया? Any doubts? Great. चलो, so we are having 5 prime to 3 prime. Yes or no? एक strand है 5 prime to 3 prime. So this will be what? 3 prime to 5 prime. Yes or no? This will be what? नीचे वाला क्या हो जाएगा? नीचे वाला हो जाएगा 3 prime to 5 prime. Clear? Okay. Great. Now see what's going to happen is that DNA polymerase 3 को लाए हम. किसको लेकर आए? DNA polymerase 3. ये कौन है हमारा? DNA polymerase 3. Okay. This is DNA polymerase 3. But again, इसके नखड़े अभी भी खत्म नहीं हुए. It said that, first of all, it said that मुझे टेम्पलेट कितना चाहिए? मुझे टेम्पलेट चाहिए 3 प्राइम से 5 प्राइम वाला. DNA polymerase 3 said that I need the template from 3 prime to 5 prime so that I can make the DNA from 5 prime to 3 prime. Okay, ठीक है, ये मान लिया हमने. Now second thing is what? This DNA polymerase 3 is saying that मुझे स्टार्ट करने के लिए कुछ चाहिए, मोटिवेशन चाहिए. जैसे तुम बोलते हो ना, सर गिव अस द मोटिवेशन, वी वांट टू गो इनटू एम्स न्यू दिल्ली, एंड डेफिनेटली वी आर गोइंग टू गो इनटू एम्स दिल्ली. बट दिस DNA polymerase 3 is also like, I can start the work but I need motivation guys, मुझे motivation दे दो, मुझे motivation दे दो. So how motivation we are going to give? He says that यहाँ पे छोटू सा double stranded लगा दो, यहाँ पे छोटू सा कुछ डबल स्टैंडर्ड का लगा दो. सो व्हाट वी हैव डन इस डबल स्टैंडर्ड को लगाने के लिए हमने किसको बुलाया? We have called an enzyme which is known as primase, and this primase enzyme what it did, एक छोटू सा प्राइमर लगाया. Now what is primer sir? Primer is a RNA sequence. So what is happening? This guy, this DNA polymerase III is a very very नखड़े वाला guy. ओके पहले तो बोला कि हमको टेम्पलेट क्या चाहिए, हमको टेम्पलेट चाहिए 3 प्राइम से 5 प्राइम. आई विल ओनली वर्क फ्रॉम 3 प्राइम. मुझे टेम्पलेट का डायरेक्शन दो 3 प्राइम से 5 प्राइम, तब जाके मैं डीएनए बनाऊंगा 5 प्राइम से 3 प्राइम. मान लिए. अब जैसे ही काम करने आया व्हाट इट सेड, I can't start work without having a double stranded thing which is primase. So primase आया enzyme, primase बेचारा enzyme RNA बनाए तब जाके ये काम करना स्टार्ट करेंगे. yes or no? तब जाके ये काम करना स्टार्ट करेंगे. क्लियर एवरीवन? ओके राइट. तब जाके ये इंसान काम करना स्टार्ट करेगा. नाउ इट इज वर्किंग, नाउ इट इज गोइंग टू वर्क. ये क्या करेगा? एक एक न्यूक्लियोटाइड पकड़ के लाएगा. क्या करेगा? एक एक न्यूक्लियोटाइड पकड़ के लाएगा. ऐसे बोलेगा तू ए है, आजा आजा. आर यू सी? आजा आजा आजा. आर यू जी? आजा आजा आजा. सो इट विल pick up all the nucleotides and finally it will make a DNA. Okay, finally it will make a DNA. Clear to everyone? Am I lagging guys? Lag हो रहा है क्या? Can dsRNA function? No. ये तो बस प्राइमर है श्रीशा. दिस इज जस्ट प्राइमर. ये बस सपोर्ट के लिए लगा हुआ है. एज सच इसका कोई फंक्शन नहीं है अभी यहाँ पे. ओके Am I lagging guys? बताना मुझे. No. Okay, great. So this is clear. Now guys, this DNA dependent this DNA polymerase 3, this DNA great this DNA polymerase 3 किसका कॉपी कर रहा है? How it is making the DNA? It's making the DNA by seeing this DNA. It has one DNA and it's copying that DNA only. If you remove the original DNA, it can't work. So can I say that this is DNA dependent? This is very good evening. मुझे तुम्हारा नाम ही नहीं समझ में आ रहा, मैं क्या बनाऊं? लायर बुलाओ तो मैं. DNA dependent DNA polymerase, yes or no? DNA dependent DNA polymerase. Can I say this guys? See, there is a DNA. तुम क्या कर रहे हो? तुम मान लो अपने फ्रेंड से कॉपी करके एग्जाम में लिख रहे हो. फ्रेंड का ही कॉपी हटा दिया कि तुम नहीं लिख पाओगे, राइट? तो मैंने क्या बोला? फ्रेंड डिपेंडेंट टेस्ट. व्हाट यू आर गिविंग? फ्रेंड डिपेंडेंट टेस्ट. सो व्हाट इट इज डूइंग? इट इज मेकिंग द पॉलीमर ऑफ डीएनए बट ऑन बिहाफ ऑफ व्हाट? किसके ऊपर बैठ के ये उड़ रहा है इतना? डीएनए के ऊपर. सो आई सेड दैट दिस एंजाइम इज डीएनए डिपेंडेंट डीएनए पॉलीमरेज. इज दिस क्लियर guys? Srishai, Shivesh, is this clear? Sanya, is this thing clear? Let me know. जल्दी बताओ मुझे if this thing is clear. Very good evening. Is it clear? बच्चों ये क्लियर समझ में आया क्या? क्लियर? ओके ग्रेट guys, great. Amin. Okay, great. Amin, I'll call you Amin. Okay, चलो. No issues. चलो. ओके ग्रेट. नाउ अब मुझे क्या पढ़ाना? देखो ये हो गया. बट नाउ सर अब यहां पे तो क्या होगा? सी हियर व्हाट इज हैपनिंग. दिस इज 5 प्राइम टू 3 प्राइम. दिख रहा है क्या? मैं ऊपर बना दूं क्या थोड़ा? इज इट विजिबल गाइज नीचे वाला? ओके आई मेक दिस हियर. आई बी लिटिल हियर. कैसे रहूं मैं? थोड़ा सा इधर हो जाऊं क्या? इधर होऊंगा तो मैं दिखूंगा नहीं. 1 second, I'll figure it out. Let me check. Am I now visible? अच्छा. मैं इधर हो जाता हूं. Okay, I'll standing here. चलो अब देखो. Now see here. So what's happening here guys? This is 5 prime and this is 3 prime. अब तो क्या? अब ये तो DNA ऐसे तो नहीं काम करेगा यहाँ पे तो ऐसे नहीं काम करेगा. Yes or no? Because ये तो बहुत नखड़े वाला एंजाइम था. It's not gonna work like this. Yes or no? अरे क्या हो रहा है ये? What's happening? हाँ ये ऐसे तो काम नहीं कर पाएगा. right? So now what's going to happen? It is going to, this DNA polymerase 3 is going to say that भाई मेरा primer लाके यहाँ लगा दो, मेरा primer लाके यहाँ लगा दो, and then I'm gonna work. Okay, so primer इधर लगा, and now again nucleotides, it is पकड़ पकड़ के nucleotides लेकर आया और इसने complete कर दिया. Yes or no? Is this thing clear? इतना समझ में आया क्या? Yes or no? This case is what? This is primer. Again, this is what? This is primer. Yes, is it clear? Chalo. Now, yes, Okazaki fragments, exactly. We will study. Now what, guys? Here tak, yahan tak to DNA replication ho gaya. Till here, DNA replication has been done, right? Till here, the DNA replication has been done. But here also we have to do, na? Here also we have to do the DNA replication. Yes or no? Yes or no? Shivesh and said, kahan bhag gaye tum? Yes or no? Yes. So, अब इसको भी तो खोलोगे. इसको खोलने के लिए फिर से एक एंजाइम लाओगे. Which enzyme? Helicase. Which enzyme? Helicase. कौन सा एंजाइम आएगा और इसको खोलेगा? Helicase. तो now see, I'm making this structure again very concentrately, very attentively. You see here, okay? क्योंकि confusion यही होता है. See, what happened was, yes, what happened was, here was what? This was the original one, okay? अभी यहाँ पे replication हुआ है. Till here, replication has been done, okay? Till here, replication has been done. Now what? I am going to open this case. I am going to open this DNA more. थोड़ा सा इस DNA को और खोलेंगे हम. With the help of helicase. With the help of helicase. So now guys, what is happening? See here. What is going to happen? ये वाला line, ये वाला DNA, तो क्या था? This DNA was, see here, this DNA was what? 3 prime to 5 prime. तो यहाँ पे बना क्या? 5 prime to 3 prime. Yes or no? यहाँ पे बन कैसा गया? 5 prime to 3 prime. But this was what, guys? This was 3 prime to 5 prime. ये क्या था? 3 prime to 5 prime था. और यहाँ पे तो DNA यहीं तक बना हुआ. Yes or no? Yes. यहाँ तक तो DNA यहीं तक बना हुआ. Yes or no? Yes or no? Now what is going to happen? ये वाला तो कंटिन्यू बन जाएगा, बट यहां पे फिर से क्या करना पड़ेगा? प्राइमर लगाओ, DNA बनाओ, प्राइमर लगाओ, DNA बनाओ. And this is going to what? This is going to be formed in the form of fragments. This is going to be formed in the form of fragments. So इस fragments को we call it as Okazaki fragments. Okazaki fragments. Yes or no? Okazaki fragments. Clear? Any doubts anywhere till here? Guys, any doubts anywhere till here? Yes, we will deal with the topoisomerase 1, 2, 3, everything. But right now is basic clear. Everyone. Sri Shai, Midlife, everyone. Amin, everyone. Is it clear? Sid? Clear? Batao, jaldi batao. If you guys have any doubts, I'll explain it again. Now see, this Okazaki fragment is what? This Okazaki fragment is a fragment. So DNA नया बनाना होगा तो भाई दोनों को तो जोड़ना पड़ेगा. You have to paste something, like you have to make a continuous strand of DNA. So इसके लिए तुम किसको बुलाओगे? इसके लिए तुम बुलाओगे DNA ligase. Why sir? Sir, okay. Now it's clear ना श्रीशाई? अब तो clear है. Now any doubt? DNA ligase. Exactly. Now we will be going to call DNA ligase. Now DNA ligase will do what? It will ligate, or I should say connect the Okazaki fragments. Connect the Okazaki fragments. Okay. Okazaki fragments. Clear to everyone? Any doubts? Anyone? Let me know in the comment section. Let me know in the comment section. I am watching the comments. बताओ मुझे. Let me know in the comment section. Thank you so much, Sid. Thank you so much, Sid. My aim is that तुम्हें कोई भी चीजों में दिक्कत ना हो. Whatever the concept I am teaching, just that concept, if you are able to grasp, my job is done. Yes or no? Great. Chalo. Now what, sir? Now I'm going to explain some of the things again. So see, this strand is what? This strand is being made continuous. In one go it is being made. Yes or no? So this is known as continuous strand. continuous DNA ligase joins Okazaki fragments. Yes, yes. SSB protein is just, you remember SSB protein, see, it's not given in the NCERT. So we don't have to remember lots and lots of things about SSB protein. You just remember that SSB protein is what? SSB protein इन दोनों को bind कर, वापस से bind करने में prevent कर रहा है. Yes or no? इन दोनों को वापस से bind करने में it is preventing. That's all. That's all you have to remember. Okay? Now see, this is known as continuous strand. Continuous strand. And it is also known as leading strand. Yes or no? पहले बन जा रहा है ना, आराम से बन जा रहा है. So it is known as leading strand. Yes or no? अच्छा, how many of you are making notes, guys? How many of you are making a proper notes of these sessions? How many of you are making proper notes of these sessions? If you are making, send that notes to me also. Okay, I'll share with the other students. Now see. Next we have what? We have the this strand. So ये क्या? छोटा छोटा थोड़ा सा हो रहा है, फिर रुक के, फिर रुक के, फिर रुक के हो रहा है. तो this is known as what? Discontinuous strand. This is known as what? Discontinuous strand. Discontinuous strand, or it is known as lagging strand. Yes or no? It is known as lagging strand. Let me know in the comment section if this is clear. Let me know in this comment section if this is clear. कहाँ send करना? हाँ, ये बात तो तुमने ठीक पूछा. Okay, I'll ask my team to create a Telegram group where we can exchange notes and all. Okay, right now though we don't have Telegram group. This is a good question, Sanya. I didn't thought about this. Okay, I'll ask my team. I'll ask my team. Okay, थोड़ा सा wait कर लेते हम लोग. And do share the channels among your friends. And let us help to hit 1000 subscribers as soon as possible, guys, as soon as possible. Okay, let us help us to grow. And if you're liking this, please, it's a request that you ask your friends and whoever is in need to subscribe to this channel. Okay, now one second sir, you told that if DNA polymerase is making a mistake, if DNA polymerase is making a mistake, then what, sir? Then it is also correcting it. Yes or no? It is also able to correct it. So ये correct कैसे करता है? See here. Just imagine guys, what's happening, that DNA polymerase 3 is working in this direction. Okay, it is working in this direction. पीछे क्या हुआ? पीछे एक चीज गलत लगा दिया. मान लो एडेनिन लगाना था, उसने ग्वानिन लगा दिया. तो ये क्या करेगा? वापस से पीछे घूमेगा, एडेनिन ग्वानिन को पकड़ेगा और एडेनिन लाके लगा देगा. So what it did? It was working from 5' to 3' direction. It is working from 5' to 3 direction. But for proofreading, it worked from 3' to 5' direction. Yes or no? Yes or no? Let me know in the comment section. Let me know in the comment section. Is it clear or not? Is this thing clear to you or not? बच्चों, clear है क्या ये बात? Is it clear or not? So can I say that proofreading, it is doing from where? It is doing from 3' to 5' direction. Proofreading, it is doing 3' to 5' direction. Clear to everyone? Clear to everyone? Shrisha said. शिवेश बताओ मुझे. Okay, I will repeat that. Okay, see again. So what I said that I'll take a blank screen. All the focus should be on me. Okay, सारा focus इधर दे दो, समझ में आ जाएगा. Okay, everyone see here. So what I was saying that this DNA polymerase 3 can make a mistake. Okay, this DNA polymerase 3 can make a mistake. So first of all, where it is working from 5' to 3' direction. Normally DNA कैसे बना रहा है? Midlife, is it clear? Midlife, is it clear? See, 5' to 3', it is normally working from 5' to 3'. So adenine लाके लगाया, guanine लाके लगाया, मान लो थाइमीन लाके लगाया. ओके, फिर ये एक एक आगे बढ़ रहा है. जैसे आगे बढ़ा, इट रिमेंबर दैट पीछे एडेनिन नहीं लगाना था, ग्वानिन लगाना था. आई हैव टू put guanine in place of adenine. So this DNA polymerase 3 has made a mistake. Now what is going to happen? It will turn back and it will go forward और उस एडेनिन को उठा के सही न्यूक्लियोटाइड लगा देगा. So when it is proofreading, when it is correcting its mistake, it's correcting its mistake in 3' to 5' direction. Is it clear? Is it clear? Normally it is working in 5' to 3' direction. Normally when the things are going well, when it has not made any mistake, it is working from 5' to 3' direction. But if it made a mistake, it returns back and correct its mistake in 3' to 5' direction. Now is it clear? Is it clear? Shri Shai, is it clear? Medlife, anyone in the comment section, is it clear? If you have any doubts, let me know in the comment section, guys. No doubts should be unturned here. Yes or no? No doubts should be unturned here. Okay. Now everything is done. Everything is done. We have to remove this primer. We have to remove this primers, guys. This primer is of no use for us. Why we have put this primer? Just because it was needed by DNA polymerase 3. So we need to remove these primers. So to remove these primers, I'm going to bring another one. PDF midlife, I will upload it in a link. After the class, like after 15 minutes midlife, you check the comment section. Here I am going to upload the link of the PDF. Okay, मैं PDF का link डाल दूंगा यहाँ पे, drive link I'll give. Okay, clear? Okay, now see what I was saying. So primer was what? Primer was of no use to us. Primer is of no use to us. We have to remove that primer. So to remove that primer, we are going to call what? We are going to call DNA polymerase 1. DNA polymerase 3 थक गया, अब वो बोला मेरे से प्राइमर रिमूव नहीं होगा भाई, DNA polymerase 1 आ जाओ. So DNA polymerase 1 is going to remove this primer and ये क्या करेगा? See here, I'll explain this. Let's suppose this is the primer. Primer was what? Primer was RNA. Yes. So if it is adenine here, so यहाँ पे क्या होगा? U होगा, T तो होगा नहीं. Thymine is present. Thymine present होता है क्या RNA में? Is thymine present in RNA? In place of thymine we have uracil. Yes or no? So here also let's suppose this is T, this is A, again this is T, this is A, just for an example. Okay, one second guys, यहाँ A कर देता हूँ, ठीक है, so that we can explain things easily. So just randomly I have written. So this DNA polymerase 1 will do what? ये U हटाएगा, साथ साथ में U के जगह पे DNA वाला T लेकर आएगा. फिर what is going to happen? It will remove A and यहाँ पे T के जगह A लेकर आएगा. It will remove U again and it will bring T again. So what DNA polymerase 1 is doing? DNA polymerase is removing the primer by removing one nucleotide, और एक nucleotide हटा के क्या कर रहा है? वो DNA वाले nucleotide लाके लगा दे रहा है. Is it clear? Is it clear? ये नो किस लिए बोल रहे तुम? ये तुम दोनों नो किस लिए बोल रहे? Let me know. Yes or no? बताओ दो मुझे. Shri Shai, Midlife, everyone, let me know. Is this clear? That is saying crystal clear. Okay. Now just 5 minutes more. Just 5 minutes more, guys. Okay. Just give me 5 minutes more. Just 5 minutes more, Midlife. Just 5 minutes more. Okay. बस एक छोटा सा टॉप बताना है चीज. Now see, this DNA replication is what? Energy लगेगी नहीं? Let me know. अच्छा. Okay. That was the question. मैं ही भूल जाता हूँ, बताऊं. Are you able to hear the crackers noise, guys? Are you able to hear the crackers noise? Okay, how many of you are going to celebrate Diwali with the crackers? When I was kid, I was very excited for the crackers, and you know, in my colony, I was the very first person who was like who used to go and bring lots and lots of crackers, and at the evening when my cracker used to get finished, what I used to do is that, like, I used to go and steal small kids crackers and we used to burn it. Acha, okay. See, now let's get back here. So DNA replication is an energy requiring process. Yes or no? Energy requiring process. So from where the energy is coming? We don't have so much ATP that can do this DNA replication. That much ATP is not present in our body. So from where we are getting the energy? See here. See, we are what? We are having the nucleotides. Yes or no? We are having the nucleotides, right? इसी न्यूक्लियोटाइड्स को क्या कर रहे हैं? यही न्यूक्लियोटाइड्स को पकड़ के लाके डीएनए का चेन बनाया जा रहा है. यस और नो? सो सी व्हाट इज हैपनिंग इज दैट दिस न्यूक्लियोटाइड इज हैविंग व्हाट? 3 फॉस्फेट ग्रुप. यस और नो? जैसे एडेनोसिन ट्राइफॉस्फेट, गुआनोसिन ट्राइफॉस्फेट. यस और नो? यस. सो लेट्स सपोज this is phosphate group, this is sugar and this is A. But when it is being used in the DNA, जब DNA में इसको use किया जाता है, so we use what? We use monophosphate. Yes or no? Adenosine monophosphate. तो ये बाकी के जो 2 phosphorus bond है, this 2 phosphorus bond is what? It is removed. And when this bond is removed, energy is generated, and this energy is what? This energy is used in the replication of DNA. Clear to everyone? So this nucleotides are serving what? Dual purpose. This nucleotide is serving as dual purpose. How dual purpose, sir? See, first though, they are what? They are providing the energy, and second, they are providing the nucleotides also. Clear? Any doubts? Sunday को भी class होती. See, this Sunday तो it won't happen, but from next Sunday it will definitely happen. Okay, this Sunday is Diwali, so if I will be able to take the class, I will take class in the morning around 11 o'clock. If not, then next Sunday we will take. On Sunday we basically have quiz and question sessions. Okay. Now this session is completed, guys. Let me know if you have any doubts. Sri Shai and Sid, anyone, Sanya, anyone, if you have doubts, let me know in the comment section. Anyone, if you have doubts, let me know in the comment section. If you like this session, please share this video among your friends, and if you have any doubts, let me know in the comment section. Okay? No doubts? Great, bacho, great. Okay. Sanya, is it clear na? Is it clear? You don't have any doubts? Is DNA replication clear to everyone? Every step, is it clear? Great. Chalo, great. So we will meet tomorrow. Tomorrow we are going to have a class at 6:30 again regularly, and tomorrow also we will be going to have those experiments, semi-conservative nature wala experiment we will have. We will solve some of the questions. So don't forget to miss that. Don't miss that class. That is also very important. And then most probably on Monday we are going to have the class. Okay. So till then, bye bye everyone.